So we've actually had 3 years in this building, which is hard to believe, but just celebrated the 20th anniversary of the Brain Research Center. So the Brain Research Center has now been subsumed into the Center for Brain Health. There was a celebration this weekend with speakers both local and international, including one who is almost guaranteed to win the Nobel Prize for his cutting-edge work. Several new labs have been brought into the Center for Brain Health, 5 labs from the Department of Psychology. That's actually relevant to this group because in particular, one of the labs, although there's plenty of room for collaboration with others, one of the labs is run by Catherine Winstanley, and there's already collaboration. There's a postdoctoral fellow, Maria Cherkasova, who's sitting right there, who works between the two places, between Catherine's lab and ours. There's 6 clinics, a lot of people, a lot of students, a lot of Canada Research Chairs. This is an extraordinary number of Canada Research Chairs, by the way, in neuroscience. 1 Canada Excellence Research Chair, who you will be hearing from later, that's Matt Farrer, and a number of endowed research chairs and donor-funded professorships. Of course, PPRI has played a major role in that exercise and a fair bit of research funding. So there are a few things that I would just like to highlight for what's happened in the past year. The Bordelin Family Biobank, which is down the hall, but don't try to get in because it's under tight security, is now receiving samples. And in fact, the Parkinson's Center is the major, not the only, but right now I think the major source of those samples. There are approximately 120 samples for the microbiome study that PPRI has been funding. And I think there are more than 200 blood and DNA samples sitting there. That's over and above the DNA samples that have been collected for many years by Matt Farrer. I just mentioned the 20th anniversary celebration that was held this weekend with these phenomenal presentations, really. And the other highlight, I think, is a partnership with the Belkin Art Gallery on campus. This is to display the drawings of Santiago Ramon y Cajal, who was a great artist as well as doctor. In fact, he wanted to become an artist, but his parents said, that's not going to pay, so you'd better become a doctor. And I'm not sure how well that paid him either. So the drawings of the nerve cells and the other cells in the nervous system are absolutely gorgeous. They are works of art. They are not just scientific. And I strongly encourage those of you who have not yet been to go to this collection, which is just across campus at the Belkin Gallery. It closes on December the 3rd, so not much time left. It has actually had more visitors than any other exhibit ever shown at the Belkin. Martin already mentioned that we hosted the Movement Disorder Society meeting here, and it was extremely successful. And I got to light the Olympic torch. So I'm going to talk about one of the programs that you have funded over the years. You're all aware that exercise is actually one of the most effective treatments we have for Parkinson's and other neurodegenerative disorders. It hasn't received the attention it deserves. Our question is not whether it works, because I think most of us are committed to the idea that it does work. The question really is to try and understand how it works. And so there are a couple of things that we wanted to look at. One is whether you could increase the release of dopamine in the brain in response to exercise, and related to that is how that works. Is there an actual change in brain circuitry? Now, when I was growing up, which was when the previous Queen Elizabeth was on the throne, we were taught that the brain was incapable of repairing itself. We now know that that is not the case, but we don't have good pharmacological treatments to make the brain repair itself, not yet. exercise, maybe. And so we designed studies that were done with your support to try and look at that. And so we had 3 aims, and I'm just going to move through this fairly quickly. We wanted to look at what would happen to motor function in the brain. But I think you're all aware that people who exercise and who actually enjoy it, I'm not a subject in this study, by the way, that they feel better afterwards. Their mental state is better. And in fact, we've noticed this, that people with Parkinson's who may have depression or apathy, this is actually better in people who exercise. And then the last thing we were interested in looking at is what is the role of inflammation in the brain in Parkinson's, and can exercise modulate that? So we have a very complicated design in which people go through an intensive exercise program 3 times a week for 3 months, and they are driven hard. Matt Sekeli, who Martin mentioned, actually did his master's degree in exercise physiology, and he was a taskmaster. And he did very detailed measurements, and I'm not going to talk about all of those, and then we had a control group. So for the first aim, we wanted to see how the release of dopamine in the brain, in the motor areas of the brain, could be modulated by exercise. In the second aim, we wanted to see whether there's a change in the way that the brain responds to reward. And then the last aim, we looked at inflammation. So we've had a lot of setbacks with this project, and I'm just going to quickly outline those. We had 16 subjects enrolled just about under a year and a half ago, and then we lost access to the PET tracers that we needed to do the studies. And what we were initially expecting to be an 8-week shutdown turned out to be around 32 weeks of lost scanning time. So we started back up in March and really cranked through. This took a lot of effort from many people, including the people at Triumph, as well as people here in the PET group. And this slide was dated in September, and at that point, there had been 70 PET scans completed by our group, of which more than 55 were related to the exercise study, which meant that everything else got shoved to the side. So we now have data on 26 subjects. In fact, Matt flew back last week from Toronto in order to complete data collection on the last 3 who were going through. And I was away, so I didn't get a chance to see him. So in order to look at what's happening in the brain, we chose to use a technique called repetitive transcranial magnetic stimulation. Now, this is used for treatment of a variety of conditions, and in fact, people have suggested that it may help Parkinson's, may help depression. We're not using this for treatment. We are purely using this as a tool to try and understand what's happening in the brain. One of my colleagues in Toronto showed several years ago that with TMS, you can evoke dopamine release in the brain. So I'll just walk you through this fairly quickly. This is a control subject, so somebody with Parkinson's, but they're in the control group, and when you stimulate the brain with TMS before they enter the study, there's no change between this dot, and they didn't release any dopamine. They were in the control group, so even at the end of the 3-month intervention, there's still no change if you compare this to this. But now let's look at somebody who was entered into the active treatment group, the aerobic exercise group. Here they are at baseline, and TMS fails to evoke dopamine release, but afterwards there's actually a change. You may not believe that by looking at the picture, so I'll show you this instead. This is dopamine release measured using PET. And what you can see is that in the people in the active group who underwent 3 months of intensive exercise, the dopamine release in response to this stimulation actually increases from a low level to a moderate level, whereas in the control group, if anything, we saw the opposite. Now, you have to take this all with a grain of salt because there's a lot of variability. But we feel this is likely a significant finding. We're waiting for the data on the last 3 people who were just scanned last week. But this would be the first demonstration of a physiological basis for the benefits of exercise, and not only that it's increased dopamine release, but it implies that there has been rewiring in the brain and the connections from the surface of the brain, the cortex, to the basal ganglia, the deeper structures that are affected in Parkinson's disease. Now, this is a very busy slide, and you don't need to worry about the details of it. This is looking at evidence of an inflammatory marker in the brain, and again, it's very early data, so I would not be definitive with this yet, but we're looking at levels of activation of inflammatory cells in the brain, and what you can see is the red bars are controls, the blue bars are people who were in the active exercise group, and there certainly appears to be a significant difference between the groups, suggesting that exercise may actually have an impact on levels of inflammation in the brain. And then lastly, for this study, we wanted to look at what happens in reward areas of the brain. So to do this, and I can tell you that response to reward in Parkinson's is not the same as in a healthy individual. So normally we actually activate the area of the brain when we anticipate that something good is going to happen. If you already know it's going to happen and then you get it, it's actually a little bit anticlimactic. So the brain doesn't actually respond to that. It's only when you think, if you think of one of Pavlov's dogs, if they know they're about to get a slab of meat, then when the light comes on or the buzzer sounds, that's when they respond actually, before the meat in fact arrives. In fact, in that case, the meat doesn't last very long once it arrives. So we designed a task in which people would play a card game, this time in the MRI scanner, and then we look at the level of activation in the brain. And then people are told they have different probabilities of winning. But what we're looking at is actually the buildup. It's that anticipatory phase. And I'll just cut to the chase here. There's a big difference between the people who are in the control group and the people who are in the active group. And in fact, the people in the active group go from a less normal response to a more normal response. So they fail to respond during anticipation here. But you can see that after the exercise program, they now have an anticipatory response, which is quite interesting because this may relate to changes in mood and apathy in Parkinson's. This is actually work that Matt presented at the Movement Disorders Society meeting, and he was one of the Blue Ribbon Highlight selectees for that meeting. But as Martin mentioned, but he was also chosen for special mention at 2 other large international meetings. One was the World Parkinson Congress in Portland last September, where the motor work that I showed you, the dopamine release work, was selected for a hot topic presentation in front of a large group. And he was also selected for special recognition at the Society for Neuroscience meeting, which has only around 35,000 people attending. We showed the same thing in a pilot study that you funded. So this is now not a controlled study. It was just looking at couch potatoes versus active people. And you can see that the active people had a much more striking anticipatory response. So this appears fairly robust, and Matt will be writing it up as we speak, I'm sure. He collected a large volume of other information on detailed motor testing, oxygen consumption, non-motor changes in the brain, and this was all done in collaboration with Dr. Lara Boyd's group. And so he's been working on some special physiological measures that I'm not going to talk about. But the reason I show this slide is because we're not dropping this now. So we're close to the end of the data collection for the first part has now being completed or nearly completed. But we want to do a long-term follow-up, and so the 3-year returns are coming back in the next few months, and we'll be following them to look at evidence for progression. But what we now want to look at is something else. So this is work that's going to be done by Michele Matarazzo, and Michele has just been in Spain. So he's not here today, I don't think. Oh, he's right there. So he just got back this morning, and I see he hasn't had a chance to shave yet. So Michele is funded by the Parkinson's Society of Canada. This is a highly competitive award. There's only one given per year, and he got it. And what we want to look at in collaboration with our colleague in Madrid is what happens to the pattern of degeneration in Parkinson's. So you probably, those of you who are Woody Allen fans will recognize this person. This is the homunculus, which is how Woody referred to the ex-husband of his current lover. And the homunculus looks distorted because the representation in the brain for different body parts is not equal. So we have a lot of representation for the tongue, mouth, and hand, and much less representation in the brain for the abdominal wall and even the leg. And that's because we require precision, sensation, and movements. So this is the homunculus in the cerebral cortex, but there is also somatotopic representation within the basal ganglia. Now, this is changed in diseases of the basal ganglia and dystonia, and we think in Parkinson's. And so what Michele plans to do is to look at the pattern of progression as Parkinson's starts and as it gets worse, what happens to the altered somatotopic representation. The other thing that we want to do that's related is to study the mechanism for dopamine release when we stimulate with TMS, because we think it's related to another population of nerve cells in the brain. So we're hoping to use PET to study that. And then finally, I just want to quickly talk about propagation of disease. With all degenerative diseases, there are proteins that become abnormally folded. So in the case of Alzheimer's disease, it's amyloid beta. In the case of some diseases, it's neurofibrillary tangles. In the case of PD, it's alpha-synuclein. So we actually decided to look at tau, which is associated with other conditions, with Alzheimer's and progressive supranuclear palsy, because it is known to interact with alpha-synuclein, the protein that's abnormal in Parkinson's. They're co-deposited in Parkinson's with cognitive impairment, and so we thought this may give us a handle on what's happening in people with Parkinson's. And so with funding from the Weston Brain Institute, we started a study, and this just shows you the difference between progressive supranuclear palsy and a healthy individual. But to our surprise, we discovered that this agent was also labeling alpha-synuclein. It's been impossible to find a good imaging agent for alpha-synuclein. This is a major area of interest for many groups. And so we may have stumbled across it. I don't think I'm going to dwell on the details here. I'll just show you that the yellow dots are high, and we expected them to be high. Those are people with progressive supranuclear palsy. But the green dots are people who have an abnormal synuclein condition, and in most of these cases, they don't yet have symptoms. They're harboring a gene mutation, and yet we're able to detect this abnormality in the brain. So this is definitely worth following up on. And I'd just like to finish by highlighting a couple of things. You may recognize this guy. He was also a Parkinson's Society Canada Fellow here and is now working in the interior of BC. So with work that actually had its initiation with support, pilot support from PPRI, and then we spent many years doing further studies afterwards. He had a paper published this year in The Lancet Neurology, which is by far the highest impact of all clinical neurological journals, has an impact factor of more than 26. So this is great We were obviously very excited about this, and we have not given up, because this young woman, who has now returned to China, was here for a year from Beijing, and we've gone back to the same journal, and I'm anxiously waiting, because the revisions, we're going to hear back about the revisions probably this week. So thank you very much for your attention.